Hello, my name is Kate Medla, and today I'm going to be sharing a video on the bacterial pathogen Clostridium botulinum. So some general characteristics are that it is a gram-positive bacteria that is rod-shaped. It is an obligate anaerobe, which means it can only survive in little to no oxygen conditions. It produces endospores as well as a bacterial toxin called the botulism neurotoxin, or shorthand BONT. There are three types of botulism. There's foodborne botulism, infant botulism, and wound botulism. And the clinical manifestations of botulism can include flaccid paralysis, problems with vision, swallowing, breathing, and difficulty in speech. So let's talk a little bit more about the neurotoxin. The structure is a 150 kilodalton protein, and it has both a heavy chain and a light chain that are linked by a disulfide bond. And this neurotoxin specifically targets peripheral nerve terminals. And so after the neurotoxin gets into your bloodstream, it finds these nerve terminals, and that's where it does this mechanism of action. The C-terminal of the heavy chain binds to the presynaptic membrane receptors of the peripheral nerve, and that is when the toxin is taken up into the synaptic endocytic vesicle. And this is when the heavy chain and the light chain separate. So the light chain is now translocated to the cytoplasm of that nerve cell, and the light chain cleaves snare proteins. So there's no vesicle fusion with the plasma membrane, and the neurotransmitter acetylcholine is not released. And acetylcholine has to do with muscle contraction. So when it is not able to be released, there will be no muscle contraction, which is the cause of that flaccid paralysis I mentioned earlier. So the light chain fragment of the cleaved toxin is actually one of the most potent toxins found in nature on this molecular weight basis. Okay, and here's just an image. So here we see that heavy chain binding to that membrane, the light chain being released into the cytosol, and then we see as the toxin gets into that endocytic vesicle, it blocks the release of that neurotransmitter and therefore inhibits muscle contraction. Okay, so pathogenesis, we're going to go over each type. So first here's foodborne botulism. So here the spores germinate in the food, and the bacteria can grow anaerobically in this food. So that's things like canned food and honey, that kind of thing, where there's no oxygen present. So the toxin is produced from these spores, and then when ingested, it is taken up by your intestinal epithelial cells. The toxin then from these epithelial cells makes its way to your bloodstream, and that's how it travels to the neurons, which is where the toxin attacks in the mechanism that I talked about in the last slide. So common sources, like I said, improperly canned, preserved, or fermented foods, and that kind of thing. Next one is infant botulism. So spores are ingested by infants, and this happens a lot with young children because they get like dirt and soil and that kind of stuff in their mouth. And so that's kind of how the spores are ingested, and they germinate and grow in the gastrointestinal tract of the infant. And so this is where the toxin is produced and absorbed by those intestinal epithelial cells. And again, the toxin makes its way from those cells into the bloodstream, where it travels to those peripheral nerves. And again, common sources, actually honey is a huge problem with the infant botulism, as well as soil and dust, like I said before. Okay, lastly, wound botulism. Spores get into the wound, again, from like soil, dust, that kind of thing. And this is where they germinate. The Clostridium botulinum grows in the wound. And here the toxin is then produced in the wound, and gets into your bloodstream. Same thing. goes through the bloodstream, gets to the peripheral nerves, and attacks. Some common sources here are contaminated drug injections or traumatic injuries like car crashes or even surgeries with contaminated medical instruments. Diagnosis of botulism. So you can get a sample from serum, feces, intestinal contents, or wound swabs, depending on which type of botulism you may have been infected with. And it can be cultured and isolated, as we can see in the picture to the right. There's also direct microscopy, so this is gram staining of smears from what you think might be a suspected food or feces from somebody you may suspect has botulism. Culture, again, here to the right, the bacteria is isolated and grown on blood agar or can be a liquid culture in RCM broth. There's the toxin neutralization test, which is also called a mouse bioassay. So you inject the contaminated food into a mouse, and if the toxin activity can be neutralized by the botulism antitoxin, then it can be confirmed that it was botulism that was in the food. And then lastly, molecular diagnosis. This can be polymerase chain reaction PCR targeted for the neurotoxin genes. Okay, treatment and prevention. So it's treated with the botulism antitoxin, but unfortunately the antitoxin does not reverse the effects of the toxin. It just stops the toxin from continuing to be produced and creating worse manifestations. Ventilator may be needed. Like I said before, botulism can cause difficulty breathing and swallowing, that kind of thing. So ventilator may be needed to help a slow return to normal breathing. Paralysis normally slowly improves. With wound botulism, sometimes surgery is needed, amputations of the infected area. And there is no vaccine for botulism, unfortunately, so we have to take lots of other preventative measures like proper storage of foods. Okay, and there are my references. I hope you enjoyed the video, and thanks for listening.